Good day, this is Professor Will, CBMD PhD. Today, Sunday, March 24, 2019. It is 9:04, 9 seconds Eastern Standard Time. This is a brief description on Archaea. So let's get started here. As microbiological techniques improved during the 20th century, microbiologists suspected that Archaea, also referred to as Archaeans, behaved in ways unique among microorganisms. In the 1970s, the American microbiologist Carl Wootz of the University of Illinois redefined microbial world by classifying microorganisms not by their outward appearance and internal structures, but by their genetic makeup. Specifically, Wootz devised a classification scheme based on the composition of ribosomal ribonucleic acid, rRNA, a component of a protein synthesis system in all microorganisms. Wootz found that Archaeans possessed some features that related them to bacteria, but other features that connected them more closely to eukaryotic cells than to bacteria. In classification schemes that divide living cells into two categories, prokaryotic and eukaryotic, the Archaea fall in the prokaryotes. That is, they lack membrane-enclosed structures, organelles inside their cells, and their deoxyribonucleic acid DNA floats freely in the cytoplasm. Eukaryotes, by contrast, are more organized cells with membrane-enclosed organelles. The rRNA studies showed Archaeans to be neither true bacteria nor true eukaryotes. The new three-domain scheme of living things, called biota, departed from the previous hierarchy of life in which all biota had been divided into 5 kingdoms, with Archaea joining bacteria and kingdom Monera. Carl Wootz and his fellow geneticist George Fox proposed their new hierarchy of life based on the rRNA studies. They wrote in 1977, "We are for the first time beginning to see the overall phylogenic structure of the living world. It is not structured in a bipartite way along the lines of the organizationally dissimilar prokaryote and eukaryote. Rather, it is at least tripartite, comprising the typical bacteria, the line of descent manifested in eukaryotic cytoplasms, and a little explored grouping, represent so far only by methanogenic bacteria, the little explored grouping, whose referred to were the Archaea." The new classification scheme prompted biologists to re-examine members of each kingdom. Science often involves the investigation of ideas, followed by debate over new findings, and then re-investigation. Few new ideas enter the mainstream of science without this scientific debate. Characteristics of Archaea. Archaea live in places that are difficult for microbiologists to sample. For this reason, few data accumulated on Archaea, while microbiology made constant discoveries on bacteria, viruses, molds, and so forth. Archaea, as do bacteria, depend on cell wall to shield them from the outside world, and their cell membrane is vital for energy-producing reactions. The Archaea's inner structure also resemble those in bacteria, but Archaeans differ from bacteria in the 4 following ways: 1 cell membrane composition, 2 cell wall structure, 3 structure and development in flagella, and 4 gene expression, which is the conversion of information carried in genes to new proteins. Archaean gene expression operates more like that in eukaryotes than in bacteria. The lipids, fat-like compounds in Archaean cell membranes differ in structure from bacteria membrane lipids. Bacterial lipids mostly contain long-chain hydrocarbons called fatty acids, which connect to a glycerol molecule. This structure allows bacterial membrane lipids to orient in a lipid biolayer. Polar hydrophilic attracted to water ends point toward the aqueous outside of the cell or toward the cytoplasm inside the cell. The non-polar hydrophobic repelled by water. Tails point into the fatty middle of the membrane. Archaean lipids, by contrast, contain 2 polar ends so that the lipids span the entire cell membrane. This arrangement enables Archaea to respond to their environment differently than bacteria or eukaryotes, which also use a lipid biolayer. Archaean membranes also contain a large, sturdy carbon compound called squalene. Squalene does not decompose at high temperatures, so it probably gives Archaea their ability to withstand very high temperatures that destroy other biological membranes. The single-layer membrane of Archaea provides more strength than biolayered membranes. A mixture of lipids containing as many as 40 carbons of specialty structures called tetraether lipids provides the durability. Tetraether lipids play a part in enabling Archaean extremophiles to thrive in the temperatures above 750 degrees Fahrenheit and 400 degrees Celsius, found in volcanoes and near steam vents on the ocean floor. Some species in domain Archaea possess peculiarities in their cell walls as well as their membranes. The stout cell wall provides protection for almost all bacteria, but some Archaea depend instead on a single S layer, a structure made of repeating proteins and glycoproteins, proteins with sugars attached to them. Archaea with S layer tend to be more sensitive to foams and other substances in the environment and act as detergent. Other Archaean species contain a cell wall similar to bacteria walls, but they differ in two other ways. First, these Archaea contain different polysaccharides, long sugar chains they found in bacteria. Second, the Archaean cell wall does not contain peptidoglycan as a bacteria, but rather a compound called pseudomoraine. Archaea with pseudomoraine react gram-positive in the identification method called the gram-stain procedure. Both peptidoglycan and pseudomoraine are composed of linkages and bridges connecting their long-chain units called polymers. In this way, both compounds provide the cell with strength. The presence of pseudomoraine in place of peptidoglycan in the Archaean cell wall explains why Archaea resists many antibiotics that kill bacteria. Many antibiotics such as penicillin and vancomycin act by interfering with normal peptidoglycan synthesis. This then leaves a bacterial cell vulnerable to damage and death. Archaea with pseudomoraine have been thought of as antibiotic resistant, but more accurately, these Archaea simply ignore any antibiotic that acts by destroying peptidoglycan. Domain Archaea holds no known pathogens to animals or plants. Archaea build their tail-like structures called flagella in the opposite manner from the bacteria used to construct their flagella. Archaea assemble their flagella from the base, rather than at the tip. Bacteria do the opposite. Once the flagella have been made, they act the same way in both Archaea and bacteria for propelling the cells in their environment. Finally, Archaean genetics share some features with bacteria genetics and have other features similar to those in eukaryotes. Archaean DNA is circular structure, as in bacteria, but the total set of Archaean genes, the genome, numbers about half of that in bacteria. Archaea additionally do nor possess plasmids, small pieces of DNA that float in the cytoplasm separately from the main DNA molecule. About 50% of Archaean genes are like those in bacteria and eukaryotes, meaning, of course, that about half of the genes are unique to Archaea. While Archaean RNA resembles bacterial RNA, the enzymes and proteins Archaeans use for replicating DNA resemble the enzymes and proteins of eukaryotes. Good day, this is Professor Will, CBMD, PhD. Today is Sunday, March 24, 2019. Well, this is the end of my brief description on Archaea. Hope you enjoyed listening. Have a great day.